In this video, we're going to talk about the adaptive immunity. Now, as we mentioned in the previous video, the adaptive immunity is characterized by three main things: specificity to a certain antigen, self and non-self recognition, and memory cells. There are two main types of adaptive immunity, depending on which cells are activated. There are B cells and T cells. If B cells are in charge of the immune response, it is known as a humoral immunity. It is an extracellular response in that it responds to microbes like bacteria. If T cells are responsible, it is known as a cell-mediated immunity. From the name cell-mediated, so it's intracellular, like tumor cells or virus, which we know replicate within a cell. B cells and T cells all originate from the same cells in the bone marrow, but T cells require the thymus for maturation, whereas B cells are released into the bloodstream when they are mature. How can we differentiate between B cells and T cells? A very important thing known as cluster of differentiation, or CD. CD marks the cell. So for example, B cell has CD19 on its surface, whereas T cell has CD3 on its surface. So when a cell has CD3, it is a T cell. There are two types of T cells. The first one is a T helper, which has a CD3 because it's a T cell, and another CD, which is CD4. CD helper cells are often called CD4 cells. The second type, which is the cytotoxic cells, CD8. Humoral immunity is basically antibodies. These antibodies are known as immunoglobulins. There are many types: IgG, IgA, IgM, IgE, and IgD. Remember, gamed. There are two types of the humoral immunity. First are the T-dependent or thymus-dependent, because we know that T cells depend on the thymus. Now remember, humoral immunity is a B cell response, but the first type depends on T cells. So let's see. This here is a T helper cell, and this antigen is a protein. Now, when this antigen binds to T helper cell along with an MHC2, which we will shortly discuss, these two will activate the T helper cell. The T helper cell will act as an antigen-presenting cell. It will bind to the B cell by certain proteins like CD40 and the co-stimulatory B7, and they will present the antigen to the B cell. When the B cell is activated, it will become a plasma cell and produce antibodies in the bloodstream. T helper cells are responsible for clonal proliferation, in that B cells will proliferate to make plasma cells, as we mentioned, which are antibody-producing cells, and memory cells, so the next time the body encounters the infection, memory cells will act rapidly. This immune response is a strong response. Unlike the second type, which is the T-independent response, meaning it does not depend on T cells, the antigen in this case is a polysaccharide or a lipopolysaccharide. This will directly activate the B cell. Instead, this response is weak. It will not form memory cells, and it will only activate IgM, immunoglobulin. This here is the structure of an antibody. Now, the antibody is made up of 4 polypeptide chains. 2 chains are heavy chains, which are the ones in blue, and 2 lighter chains, which are the orange chains. Now, this region here, which binds to the antigen, is known as the variable region. It varies according to the antigen it binds to. This area here is known as the Fc region. This region binds to natural killer cells, neutrophils, eosinophils. Let's take an example. Let's imagine that this is a bacteria displaying a surface antigen. Now the antibody will bind to the bacteria, and this region, which is known as the variable region, is specific to this antigen. The Fc region is going to recruit white blood cells, and this will aid in the engulfing process. In primary infection, the first response is usually IgM, whereas secondary response has a more potent immunoglobulin, which is IgG. The primary response is longer and weaker. Long meaning it takes a long time for IgM to develop, and the response is weak. IgG is quick because IgG is already available from the memory cells, and it is a stronger response. The second type, which is the cell-mediated immunity, depends on T cells, as we said. The two types, which are CD4 and CD8. CD4 has two types, T helper 1 and T helper 2. T helper 1 is responsible for activating CD8 cells, which later activate macrophages. T helper 2 activates B cell and, as we mentioned, acts as an antigen-presenting cell. The cytotoxic, from its name, it's a cytotoxic, so it kills the cell. It kills tumor cells, viruses, and allografts. Plus MHC will activate T lymphocytes and initiate a response. We already talked about what an antigen is, and we know what T lymphocytes are. What are MHC, and what type of response do they give? MHC is major histocompatibility complex. There are two types of MHC, MHC class 1 and MHC class 2. MHC class 1 is composed of HLA-A, B, and C. MHC class 2 include HLA-D, and there are many types like HLA-DR, DP, and DQ. So let's imagine that this here is chromosome 6. The short arm of chromosome 6 will have certain genes. These genes are known as human leukocyte antigen genes, HLA. HLA genes are clustered to make MHC complex. Now, MHC class 1 is found on all nucleated cells. The main goal is to activate CD8, and CD8 will eliminate the host cell itself. MHC class 2 is presented on antigen-presenting cells, and they help the T helper 2 cells present the antigen. As we said, when MHC and the antigen bind, they will activate T cells, and T cells will produce cytokines. The two most important cytokines are interleukin 2, which help the T cell grow, produce memory cells, and activate CD8. The second cytokine is interferons. Interferons help macrophage activation. So when the macrophages and the CD8 are activated, they promote cell killing by the perforins are proteins that create holes or pores in a membrane. Granzymes are enzymes that break up the membrane, like protease, and cytokines which induce cell death or apoptosis. Thank you for watching the summary on adaptive immunity.